To start this presentation, we need to know what antioxidant is. Well, even from studies, antioxidants are difficult to define. In literature, the term is restricted to chain-breaking antioxidant inhibitors of lipid peroxidation. In broader terms, an antioxidant is any substance that, when present at low concentrations compared to those of an oxidizable substrate, delays or prevents oxidation of that substrate. So, how do we determine the effectiveness of antioxidants in the body? This is by looking at the antioxidant capacity or activity. Antioxidant capacity is the overall ability of a sample, such as food or plant extract, to neutralize free radicals or reduce oxidative stress. As for the antioxidant activity, it refers to the ability of a substance to inhibit or delay the oxidation of a target molecule, thereby protecting it from oxidative damage. Factors such as concentration, chemical structure, and reaction kinetics can influence antioxidant activity and capacity. First, the higher concentrations of antioxidants exhibit greater antioxidant activity. Second, different antioxidant compounds have varying antioxidant capacities due to differences in their chemical properties and reactivity with free radicals. Third, factors such as rate constant, reaction mechanism, and overall reaction efficiency can impact the effectiveness of an antioxidant in neutralizing free radicals. So, why do we need to do analysis for the antioxidant activity and capacity? What is the importance? Well, there are many benefits to this analysis for sure. By analyzing the antioxidant activity and capacity, we are able to assess the health benefits against oxidative stress-related conditions. Other than that, we are able to compare and rank samples based on the antioxidant contents and efficacy. Moreover, it is possible to develop antioxidant-rich products. And last but not least, we can understand the mechanism of action of antioxidants, which is vital for elucidating the molecular pathways and interactions involved in antioxidant defense system and oxidative stress modulation.
Next, we focus on the method that we will use to determine the antioxidant activity. First method is 2,2-diphenyl 1-picril hydrazine, or we known as DPPH. What is DPPH? DPPH is formed on dark color crystalline powder and is composed of stable free radical molecules. DPPH is popular radical and a very efficient scavenger for other radicals, and it used to monitor chemical reaction that involve radicals, which is antioxidant assay. Next, the principle of the DPPH method. DPPH is a popular, quick, easy, and affordable approach for the measurement of antioxidant properties that includes the use of the free radicals used for assessing the potential of substance to serve as hydrogen providers or free radical scavengers. It is used to measure the antioxidant activity of molecules which have the ability to transfer electrons and make a reaction with organic radicals. The free radical DPPH interact with an electron to yield a strong absorbent at 517 nanometer. Mechanism by which DPPH accepts hydrogen from antioxidants. Initially, DPPH, a free radical solution in methanol, is a dark purple color, and antioxidants donate the hydrogen atom to the DPPH. Hydrogen form a bond with the nitrogen because DPPH is very reactive. When hydrogen was accepted, the dark purple color disappeared. The intensity of the dark purple color slowly decreased and the absorbance also decreased. The color was changed from dark purple to yellow. Next, antioxidant activity is inversely proportional to absorbance. That means more the antioxidant property, less will be the absorbent. And if there is no antioxidant, the absorbent will be higher and form dark purple color.
Next method is 2,2-azinobis 3-ethylbenzothioazolene 6-sulfonic acid, or also known as ABTS. So, what is an ABTS? ABTS is used to determine the total antioxidant capacity of plant, extract, food, and clinical fluid. It is commonly used as a substrate in biochemical assays, enzymatic kinetics, and ELISA experiments. And ABTS widely used in different research areas such as food science technology, agriculture, plant science, and nutrition. Moving on, the principle of ABTS. First, ABTS is further converted to its radical cation by adding sodium persulfate. Second ABTS radical cation is the chromophore, which is actually blue in color and has absorption maximum at 734 Nm. Third, the chromophore that form is reactive towards many antioxidants like phenolates, thiols, and vitamin C. Fourth, the antioxidant reduce the ABTS, which depends on the activity and the concentration of the particular antioxidant. And the fifth, blue color of ABTS radical cation chromophore reverts to its colorless neutral form. The last method is beta-carotene bleaching assays. What is beta-carotene and beta-carotene bleaching assays? Beta-carotene is an organic strongly colored orange pigment abundant in fungi, plants, and fruit. Beta-carotene is an antioxidant and it protects the body from damaging molecules called free radical. Beta-carotene bleaching assays determines the capacity of the antioxidant to inhibit lipid peroxidation in the phase of initiation as well as in the phase of propagation. Moving on to the principle of beta-carotene bleaching assays. First, it based on the loss of the yellow color of beta-carotene due to its reaction with radicals which are formed by linoleic acid oxidation in an emulsion. Second, the method works in an aqueous emulsion of linoleic acid and beta-carotene, which is colored by the radicals generated by the spontaneous oxidation of the fatty acid promoted by thermal induction, typically at 50 degrees Celsius. Third, the absorbance of the emulsion was determined at 470 Nm every 15 minutes until 120 minutes. And lastly, the fourth beta-carotene was oxidized by the free radicals produced from oxidized linoleic acid, which more closely simulates a real food system compared to DPPH and ABTS methods.
Next, the sample used to determine the antioxidant activity. In this video, we use broccoli and asparagus. This is because the antioxidant activity of broccoli was ranked as the second in 10 of common vegetables. Next, asparagus. Asparagus shows one of the greatest antioxidant activities in 43 vegetables, and it rich in number of bioactive compounds such as glutathione, vitamin C, vitamin E, and fructans. Preparation of the samples. Step 1. Samples were chopped to small pieces. Step 2. The macerate was homogenized with 10 ml methanol, acetone, or water. Step 3. The clear supernatant was transferred to a plastic bottle and stored. Step 4. Samples were wrapped in the 3-layer of cheesecloth and hand-pressed to prepare the juice. Last step, the crude juice was centrifuged and the clear supernatant was collected for analysis.
Analysis, the antioxidant activity. Measured by DPPH method, the antioxidant activity of asparagus was significantly higher than broccoli, and the antioxidant activity of methanol extracts were higher than acetone and water extract. As measured by the ABTS method, antioxidant activity of asparagus and broccoli did not show significant differences, and antioxidant activity of extracts using the 3 solvent did not show significant difference either. In beta-carotene bleaching assays, beta-carotene was oxidized by the free radical produced from oxidized linoleic acid, which more closely stimulates a real food system compared to DPPH and ABTS methods. According to the beta-carotene bleaching method, there was no significant difference in the antioxidant activity of the 2 vegetables, but acetone extract had higher antioxidant activity than those of methanol and water extracts. From the present experiment, methanol and acetone extract had higher antioxidant activity than water extracts, possibly because flavonoids are major antioxidants in both vegetables and are more soluble in methanol and acetone than water. Thus, water alone is not a good solvent to extract the antioxidants of asparagus or broccoli. In summary, asparagus had greater antioxidant activity than broccoli, and methanol and acetone extract showed greater antioxidant activity than water extract. There was no significant difference in total phenolate content between asparagus and broccoli, but asparagus had higher contents of flavonoid than broccoli. The antioxidant activity demonstrated significant relationship with flavonoid content in asparagus and broccoli. It is recommended to use more than one method to evaluate the antioxidant activity of asparagus and broccoli, as each assay has its own mechanism.